Thank you, Dr. Leguda, for the introduction, and also thanks to the organizers for inviting me to this wonderful venue to present some of the latest results from our lab. Now, the title of my talk is Segregation for Aggressiveness in Sexual Offspring of the Aeroros pathogen Puccina streiformis. And first, I would like to acknowledge PhD fellow Julian Rodriguez Algaba for generating the sexual offspring that I have been using in this study. And if you want to know more about how to generate offspring, you can go and visit his poster here at the workshop. And also, this study was done under the supervision of Professor Måns Hovmøller and senior scientist Anne-Marie Justesen. So today, I would like to give you a brief introduction to the definition of pathogen aggressiveness and some of the important questions that we need to address. Then I will give you a short introduction also to the experimental design and setup that we have developed for assessment of aggressiveness of Puccina streiformis, and then the results we have for segregation of aggressiveness in the progeny isolates, and in the end some conclusions. So if you look in the literature, you can find many different definitions of pathogen aggressiveness. And some of them can be quite complicated. But the one I put forward here is the one I think is the most straightforward one without being too simple. And it is described as the relative ability of a virulent isolate to cause disease on a susceptible host. Most often, pathogen aggressiveness has been assessed by looking at some epidemiological components like germination rate, infection efficiency, latent period, lesion growth rate, and spore production. And of course, all these components, they are quantitative characters. And as you can see in the headline, pathogen aggressiveness is also sometimes described as the quantitative part of pathogenicity as opposed to virulence, which is a more qualitative way of assessing the interaction with the host. So since these parameters are quantitative parameters, they can be quite difficult to handle, because often you see a lot of variation in your data, and also you can have a large effect of environment. So ideally, you would like to do your experiment in a very controlled environment. Now, some of the important questions then you can ask in relation to pathogen aggressiveness is the first one here. Is there actually genetically based variation for aggressiveness in pathogen populations? And we don't have many data on this, particularly not for Aeroros, but if we look at some of the recent examples of spread and invasions, we have indications that we have variation for aggressiveness. For instance, the discovery of the aggressive and high temperature adapted strain in the US in year 2000, which then later on spread and caused severe problems in East Africa and also here in Australia. That's a good indication that something is going on. And then the second question, do we actually see segregation of these traits when the pathogen goes through sexual reproduction? Now, this question is, of course, very important in order to understand how sexual reproduction are able to shape populations. And then the last question here, do we see genetic correlation between aggressiveness traits? Are there any possible trade-offs between any of these traits that can prevent the formation of super aggressive genotypes? And again, we don't have much data for this, but it has been shown a few times for leaf rust or the Puccina triticina that you actually see that those isolates that has a short latent period, at the same time they don't produce as many spores as those with the long latent period. So in the study, we used some progeny isolates that came from a selfing of an isolate of the so-called warrior race. This race was first detected in Europe in year 2011, and already in the first year of detection, it was very prevalent. And as you can see here on the graph, it is represented by the pink color. Now since then it has been the dominating race in Europe. One of the important characteristics of this isolate or this race is that it has a readily production of teliospores, and that's of course an important basis for doing sexual reproduction. And this again also separates it a bit from the old European population where you don't see a readily production of teliospores. So this progeny isolate that I used in this study, we have already some published results for these progeny isolates. It was published last year in Fungal Genetics and Biology. And in this study, we were able to confirm the parental origin of the progenies based on SSR markers. And we also saw segregation for SSR markers. So in a total of 16 progeny isolates, we had 11 multilocus genotypes. And there were also a few indications of segregation for virulence. So in this study of segregation of aggressiveness, we developed a system for how to assess the aggressiveness parameters. For inoculation, we fixed the second leaf of seedlings on plexiglass sheets, and then we did a semi-point inoculation with spores suspended in an engineered fluid. Now the beauty of this system was that you could actually evaluate different isolates on different leaves side by side within a very small tray, so we could keep all the treatments we had in a very small environment, so that they were all the environment was very controlled. Then after inoculation, the trays were covered with a lid and left for incubation in 24 hours. And after incubation, we moved the trays to quarantine spore-proof cabins with an automatic water system and left them there without touching them for about 8 days until we started the scoring. So we had two fixed factors in this experimental design. We had variety and we had isolates. The variety, they were two susceptible varieties called AVOZS and 514W. In the 514W, we have no known resistance, and there's a resistance gene in AVOZS, but it's only effective against a very, very limited number of the known races. Of the isolates, we had, of course, the parental isolates. Then we had 17 progeny isolates. And then we had two reference isolates that we had some knowledge about from previous studies. We had an isolate that belonged to the aggressive strain that was first discovered in US in year 2000. And then we had an isolate of the old European population. So, and all these variety isolates combinations, they were replicated 12 times. And the whole experiment was replicated 3 times at 3 different times of the year. So we had a total of 1,440 inoculations with a success rate of 96.5%. And the two parameters that we looked at in this study was latent period and lesion growth. And I will show you how we did the assessment. So for latent period, latent period was considered as the time from we did the inoculation until we saw the first open pustules on the leaf. And then 4 days after seeing the first open pustules, we marked the edges of the lesion by a filled pen. And then again, 4 days later, we marked the lesion edges again, and then the difference between the two markings was considered as the lesion growth. So of course, we were not really interested in the effect of experiment in this study. We just did 3 replications of the whole experiment to kind of generalize the study. So the data was analyzed in a mixed model with experiment as a random factor. And both for latent period and lesion growth, we had a significant effect of variety and also a significant effect of isolates. But for lesion growth, we also saw a slightly significant effect of variety-isolate interaction. So I split the analysis based on variety. And here you have distributions of the LS means that was generated from the mixed model. And to your right, you have the distribution of latent periods. And to the left, you have the distribution of, sorry, the other way around. To the left, you have the distribution of latent period, and to the right, you have distributions for lesion growth, and split on 2 different hosts, the AVOZS and the 514W. And on the x-axis, you have all the isolates, and then on the y-axis, you have the values for latent period and lesion growth. And the black bar somewhere in the middle of the distribution represents the parent isolate. So interestingly, we had some conserved patterns of the distribution on both varieties. Four isolates, they had a longer latent period than the parent on both varieties, and in some cases the differences were significant, as marked by the asterisk here. And when I say that they were significantly different, it means only significantly different from the parent, because there are of course more significant differences if you look at the whole data. And interestingly, the same 4 isolates, they also had a faster lesion growth than the parent. But if you look at the other end of the spectrum, then we had 4 isolates that had a slower lesion growth than the parents, but at the same time they had a latent period that was either equal to the parent or slightly shorter. So of course these distributions, they show that we have indications of segregation for aggressiveness in these progeny isolates. But also, they also indicate that we have a trade-off between lesion growth and latent period. Because those isolates, just show here, those isolates that had a long latent period, they at the same time had the fastest lesion growth. So I went on to look at the correlation between the latent period and the lesion growth. And again, they were split between varieties. So if you look at the correlation here, and you have the latent period on the x-axis, and you have the lesion growth on the y-axis. And all the squares, they represent the LS means for each isolate on the variety 514W, and then the triangles, the LS means on average S. And as you can see, we have a very nice correlation on both varieties, and those correlations are of course significant. But intuitively, they also look parallel, those 2 lines. But just to confirm that, I did an analysis of covariance, just to see if there was any interaction between latent period and variety on lesion growth, and we didn't find any significant interaction. That indicates that the correlation between latent period and lesion growth was similar on both varieties. But coming back to this scatter plot here, just by looking at it, we can see that the LS means they are very nice, or the isolates, they are very nicely scattered out. And if you go into more details, we can see that there are also some groupings. We have the first group here, which is centered close to the parent isolate. And then we have a group up here marked by the red. And those were the isolates that had a long latent period, but at the same time a faster lesion growth than the parent. And they are the same, of course, the same isolates on both varieties. And somewhere in between, we had also the same isolate on both varieties. And then lastly, we had a group down here. And those were the 2 isolates, and again the same on both varieties. They had a slower lesion growth, but a latent period that was kind of comparable to or slightly faster than the parent. Now it starts to look a bit crowded, the graph, but I would like to just mark one more isolate. And it's the isolate here, marked in green, that's called A22. It was not found in this study to be significantly different from the parent. But there was indications that it was more aggressive than the parent in the sense that it had both a shorter latent period, but also a faster lesion growth. And I'm sure if we had studied some other parameters, like for instance disease severity, we would have found that this isolate could cause more disease than the parent. And one more interesting thing about this isolate was that it was associated with a very special phenotype. So if you took all these isolates and you inoculate them onto seedlings, then after a few weeks, all the isolates, they will start to produce cilia, but not this isolate. It seems to have lost the ability to produce cilia. And this is just speculations, but one of the kind of important theories in the theory of evolution of pathogens is that there's a kind of fitness penalty by doing sexual reproduction. And this goes very well with this isolate here, so because if it has lost the ability to produce cilia, it cannot go through sexual reproduction, but at the same time it seems like it's more aggressive than the parent. it's of course speculation, but it's it's a nice observation anyway. Also, sorry, just go back. Also, there was also a nice phenotype associated with the 4 red isolates. If we looked at should have been a little different, but anyway, if we looked at the infections 12 days after inoculation, you could see that those 4 isolates, they were associated with chlorosis on the leaves, and this was the case on both hosts. I hope you can see it on the picture. But then again, if we looked 1 week later at 19 days after inoculations, you could see that all the interactions, they were clearly compatible. And if you looked in the new part of lesions for those red isolates, then we didn't observe any chlorosis. So the chlorosis were only present in the initial infection sites. Yeah, then just a few conclusions about the study. We developed a system for simple and repeatable assessment of components of aggressiveness for Porcinus staphylococcus on wheat seedlings. And this seemed to work very nicely with the point inoculations and the assessment of latent period and lesion growth. It was quite easy to handle. Then we had evidence of segregation for both latent period and lesion growth in even a very small set of progeny isolates that came from a selfing. And then we had the indication of a trade-off effect between latent period and lesion growth. And in the end, similar results were obtained on 2 susceptible wheat varieties with 2 very different genetic backgrounds, indicating that the effects that we saw was mainly due to the genetics of the isolates and not so much related to interaction with the host. So just one perspective I would like to put on these results is that we all know that the use of major resistant genes in the field can lead to selection for virulence. But I think since now that we know that we have variation for aggressiveness in pathogen populations, but also that when the pathogen goes to sexual reproduction, we actually generate more variation for aggressiveness, we should try to study more how this affects the use of partial resistance in the field, and especially partial resistance in areas where you have sexual reproduction. Because you might risk selecting for aggressive genotypes, for genotypes that's more aggressive against varieties that carries partial resistance. So some acknowledgements. I would like to acknowledge all my colleagues at the Global Rust Reference Center, in particular Professor Måns Haumøller and Senior Scientist Anne-Marie Justesen. I would also like to acknowledge our partners at INRA Institute in France, Dr. Claude Pope and engineer Marc Lecomte, for a discussion on how to assess pathogen aggressiveness. Financial support came from the Innovation Fund Denmark to the Rust Fight project, and then of course the Aarhus University. Thank you for your attention.